Today we focus on the periodontium. You know that periodontium comprises the structures responsible for maintaining the teeth in the maxillary and mandibular bones and includes the cementum, the periodontal ligament, and alveolar bone with its gingiva, associated gingiva. And here on this slide, on this picture, you can see the components of periodontium, which consist of supporting soft and hard dental tissues between the tooth and the alveolar bone, as well as parts of the tooth and alveolar bone. Yes, here you can see this alveolar bone, and also you can see the periodontal ligament and the cementum, and also gingiva, which covers the periodontal ligament and the alveolar bone. As we are going to discuss cementogenesis, we have to recall the root development and the root formation of teeth, of dental development. Root development occurs after the crown has commenced its development and involves interactions between the dental follicle, the enamel cervical loop of the enamel organ, and its extensions, the epithelial root sheath. Soon after the crown has completed its formation, the external and internal enamel epithelia at the cervical loop of the enamel organ form a double layered epithelial root sheath, you can see here, and the cells of inner enamel epithelium form this sheet of Hartwig's, and it remains short and induces the undifferentiated mesenchymal cells of the dental papilla to differentiate into odontoblasts that secrete dentin. So the function of the root sheath is to mold the shape of the root and initiate dentin formation. This double layered epithelial root sheet, which is called also Hartwig's root sheet, it proliferates apically to map out the shape of the future root. The primary apical foramen at the growing end of the epithelial root sheet may subdivide into a number of secondary apical foramen by the ingrowth of epithelial shells. Here you can see shells from the margins of the root sheath that subsequently fuse near the center. Here you can see this fusion near the center of the root. The number and the location of these epithelial shells correspond to the number and the location of the definitive roots of the tooth and may be under the inductive control of the dental papilla. It has been suggested that the ingrowth of the epithelial shells takes place along paths of low vascularity. And here on this picture, you can see the epithelial root sheath and tissue morphogenesis. And here you can see this two-layered Hartwig's epithelial root sheath. And during root development, growth of the epithelial root sheath occurs to enclose the dental papilla, except for an opening at the base at the apical foramen. Beneath the dental papilla, the epithelial sheath usually appears angled to form the root diaphragm. Here you can see, yes, this is the root diaphragm. Unlike the cervical loop region in the enamel organ of the crown, there is no stellate reticulum and stratum intermedium between these two layers. Here, between these two layers, there is no stellate reticulum and stratum intermedium. between these two layers of the root sheath, as the cervical loop region is the site of the stem cell niche. Its replacement by the epithelial root sheath may be related to the disappearance of notch protein in the epithelium and growth factors. Fibroblast growth factor and bone morphogenic factor in the adjacent mesenchyme. The localized retention of such factors may help explain the occasional presence of stellate reticulum and stratum intermedium in the root, giving rise to a rise of enamel pearls on the root surface, and we'll see a bit later these pearls. The dental follicle lies external to the root sheath and forms cementum, periodontal ligament, and alveolar bone. And today we discuss these three structures and their development. In the region of root diaphragm, here, this root diaphragm may be here, this is root diaphragm. This epithelial root sheath is seen as a continuous sheet of tissue sandwiched between the undifferentiated mesenchyme of dental papilla and internally, and the dental follicle externally, and separated from both by a basement membrane. And here you can see this basement membrane from basement membrane, and above the root diaphragm towards the developing crown, the cells of the internal layer of the epithelial root sheath induce the peripheral cells of the dental papilla to differentiate into odontoblast, and here you can see the differentiating odontoblast at different stages of differentiation. Following the onset of the dentinogenesis in the root, the epithelial cells of the root sheath lose their continuity, becoming separated from the surface of the developing root dentin here, and the mesenchymal cells of the dental follicle adjacent to the root here, they these cells near root dentin, they now differentiate into cementoblasts. Here you can see these are cementoblast-like cells, and the cementogenesis commences with the formation of acellular primary cementum. There is evidence, however, that epithelial cells of the sheath may also differentiate into the first formed cementoblast-like cells. The epithelial remnants here you can see are retained and subsequently form the epithelial rest cells. Here you can see these some islands which are called epithelial cell rests of molasses. And in the periodontal ligament here, this is developing periodontal ligament. This micrograph illustrates the apical region of the developing root, periodontal ligament, and dental papilla, and alveolus. Here you can see the alveolar bone development, and the tissues of the dental follicle in the developing root have been described and comprising three layers. Here you can see these three layers between between the alveolar sheath and between the alveolar bone, and this tissue of the dental follicle in the developing root have been described as these comprising three layers, as we mentioned, adjacent to the epithelial root sheath. Here, epithelial root sheath is the inner investing layer of the dental follicle, which is thought to be derived from ectomesenchyme neural crest cells here. Adjacent to the developing alveolar bone here, alveolar bone near. It is outer layer of dental follicle, which is separated from the inner layer by an intermediate layer here, intermediate layer. And unlike the tissues of the inner layer, these outer and intermediate layers are said to be mesoderm in origin. Their cells contain few cytoplasmic organelles, and the extracellular compartment appears relatively featureless. Cells of the inner layer of the dental follicle differentiate into the cementoblasts. You can see here developing here you can see developing cementum and cementoblasts, and that form an initial layer of cells on the surface here, this surface or initial layer of the root dentin. And in primary acellular cementum, the collagen is of the extrinsic fiber type, being derived from the periodontal ligament and passing into the cementum roughly perpendicular to the surface of the root. The cementoblasts associated with acellular cementum therefore contribute little material towards the extracellular matrix of the tissue. Later, with the formation of cellular secondary cementum, the cementoblasts form a more distinctive layer of cuboidal cells. Here you can see these cuboidal cells that secrete collagen parallel to the surface, forming the matrix of the cementum, intrinsic fiber cementum. Once cementogenesis has begun, cells of the remaining dental follicle become obliquely oriented along the root surface and show an increased content of intracellular organelles, becoming the fibroblasts of the periodontal ligament here. And these fibroblasts secrete collagen of the periodontal ligament. This part gives rise to periodontal ligament and into the extracellular compartment. This collagen will become embedded as sharpie fibers into the developing acellular cementum at the tooth surface and into the developing bone at the alveolar surface. Here at the alveolar surface from this periodontal ligament. There has been controversy concerning the connective tissue immediately beneath the developing root apex, and here you can see this. Initially, it was called cushion hammock ligament, and this connective tissue was described as a fibrous network with fluid-filled interstices, with attachment on either sides to the alveolar wall. It was thought to provide a resistant base so that forces produced by the growing root were prevented from causing bone resorption and were resolved into an eruptive force. But now this view is no longer held, as the thin fibrous membrane seen in this site is not attached to alveolar bone, but merges at the sides with the fibers of the developing periodontal ligament. The structure is more correctly termed pulp-limiting membrane, and it does not appear to be directly involved in tooth eruption, as its surgical removal does not affect tooth eruption. Here you can see a section of developing root of extracted tooth, and you can see this epithelial root sheet. This is dental papilla, and at the superficial layer you can see the differentiating odontoblast at the periphery of the dental papilla, and also you can see the matrix of mineralized dentin. And here you can see unmineralized predentin, here unmineralized predentin, and also you can see the dental follicle around this enamel organ, and which will give rise to the periodontal ligament. And here you can see that this was the multiple injected, this tooth was a multiple injected of the antibiotic tetracycline. The antibiotic was incorporated into the tissue mineralizing at the time of the injection, and this we like appearance, yellow appearance, you can see, and there is continuity at the edge of this section where the original predentin recurves upwards at the side of the hyaline layer. And here you can see the section with this picture shows the an erupting permanent molar, permanent molar into the oral cavity, emerging into the oral cavity, and you can see that there are very clearly defined periodontal ligament fibers, and here it is erupting second premolar of squirrel monkey just emerging into the oral cavity, and you can erupt it first premolar, and there is absence of periodontal ligament fibers. Prior to root formation, the cells of the dental follicle have two characteristics, the characteristics of undifferentiated fibroblasts, containing few cytoplasmic organelles. With the onset of root formation, the cells show an increase in cytoplasmic organelles, especially those associated with protein synthesis and secretion, and extracellular space begin to fill with collagen and ground substance. These cells of dental follicle give rise to cementoblasts, fibroblasts, and osteoblasts of the periodontal ligament. There is evidence that cells from the apical region of the dental papilla migrate around the epithelial root sheath and supplement the number of cells in the dental follicle. Cells may also migrate into the developing periodontal ligament from the endosteal spaces of the adjacent alveolar bone. During the formation of the root and periodontal ligament, there is obvious a dramatic increase in the number of connective tissue forming cells such as cementoblasts, fibroblasts, and osteoblasts. Stem cells exist within the region to allow for this, perhaps lying in a perivascular location, and it is not clear whether the three main cell types all arise from a common stem cell or whether each type has its own specific stem cell. There appear to be few structural differences between fibroblasts in the developing periodontal ligament of erupting teeth and those associated with fully erupted tooth. In addition to the main connective tissue forming cells, osteoclasts also appear in the developing periodontal ligament at the alveolar bone surface, allowing bone to remodel an association with tooth eruption and growth. As the tooth erupts, there is little evidence of any significant bone deposition at the base of the sockets. Uh, Here you can see the collagen fibers which are embedded into cementum on one side and the periodontal space in the and into alveolar bone in the other side. This side is from right, it is alveolar bone. The embedded fibers are termed as Sharpie's fibers. Here you can see the formation of periodontal ligament fibers. At the start, you can see at the start, these short tufts of fibrils are formed at the bone surface. This is bone surface, and at the cementum here, at the cementum surface, collagen fibers still lie parallel to the bony wall in the periodontal space. The fiber bundles close to the bone close to the bone first grow more quickly, here you can see, and branch until they approach the fiber bundles on the root side, and together they form the mesh of periodontal hair fibers. These essential cells, osteoblasts, cementoblasts, osteoclasts, and fibroblasts, reside here on the surface of this alveolar bone and in from the on the side surface of cementum, and they also you can find here blood vessels. Blood vessels in the forming periodontal ligament seem to be derived from two main sources. They enter from the periapical area, pass upwards within the ligament, and form anastomosis with vessels entering from the adjacent developing alveolar bone. As the tooth erupts, it receives vessels from the adjacent gingiva. In the case of permanent teeth, connections also develop with the periodontal ligament of the overlying deciduous teeth. There is evidence that differences may be present in the periodontal vessels of erupting and fully erupted teeth. In that the number of fenestrated capillaries is greater in erupted teeth. And the next slide, you can see this next slide, which indicates this is histological section of bone graft interface, and you can see the Sharpies like fibers, and which anchors the soft tissue graft to the bone tunnel. And here you can see the periodontal ligament and alveolar bone from left side, and dentine from cementum and dentin from the right side, and you can see the Sharpies fibers which penetrate the bone as well as the cementum. And here you can see the mineralization front in the developing root extending from the dentin, and you can see these are Sharpies fibers. This is cementum, and here you can see dentin cementum junction, here dentin cementum junction, and also you can see developing dentin here, and this is the fibrous fringe of the cementum. This you can see this cementum, and also you can see the cementoblast on the left side. You know that cementum is a mineralized connective tissue. It is not vascularized or innervated, and it covers the outer surface of the tooth root in a thin layer. The fibers of the periodontal ligament insert into the cementum. In the cementum, these fibers are known as Sharpie's fibers. The periodontal ligament fibers attach the teeth to their bony sockets, alveolus. This is why cementum is also part of periodontium. Cementum was first described in 1835 by pupils of the physiologist Purkinje in Prague. And a distinction is now made between five different types of cementum in humans. These types differ with regard to structure, fiber, and cell content, but also in terms of localization, rate of formation, biochemical composition, and degree of mineralization. In cementum, normally requires dentin as the base into the which it is deposited. Human dental enamel is free of cementum deposits, which is why a cement enamel junction can be established at the neck of the tooth. Variations occur in which a few cemental spurs extend beyond the cemental enamel junction and lie on the enamel. A few insular cementum deposits can also be found on the enamel. The formation of cementum is closely associated with the development of the tooth root. After formation of the crown is complete, the inner and outer enamel epithelium face each other. Here, maybe, yes, here, may face each other, and this duplicature of the epithelium is initially known as cervical lobe, and from it the Hertwig's epithelial root sheath emerges, which defines the shape during root formation. This involves continued cell division in the apical region while the coronal portion disintegrates. The cells of the Hertwig epithelial root sheath form a circumferential surface onto which the first deposits of root dentin are apposed. In this way, it shapes the root. With advancing root formation, the Hertwig epithelial root sheath migrates slightly further in the apical direction. Coronally, it continues to disintegrate, but not entirely. As loosely woven mesh of epithelial strands persist, and here you can see, and thins out ever further. This is referred to as epithelial rest of molasses. Here you can see the region around the root apex, which is just forming, is rich in ectomesenchymal progenitor cells, from which fibroblasts, preodontoblasts, and pre-cementoblasts are derived. The epithelial cells from the Hertwig epithelial root sheath can act on the progenitor cells, making use of the close proximity. Thus, preodontoblasts, among other cells, originate from the progenitor cells. They secrete polypeptides that are similar to enamelin and amelogenin. This process has not been detected for cementoblasts. In cementogenesis, the Hertwig's epithelial root sheath disintegrates enough to enable the dentin surface of the freshly formed root to come into contact with cells of the dental follicle. This is thought to initiate the differentiation of connective tissue cells from the dental follicle into cementoblasts. At any rate, deposition of cementum onto this dentin surface starts as soon as freshly formed surfaces of root dentin are freed out of their covering with the Hartwig's epithelial root sheath. As yet, it is not known what influences initiation of the differentiation of dental follicle cells into cementoblasts. There is also debate about whether any cells of the Hartwig epithelial root sheath become cementoblasts. If so, this would necessary an epithelial mesenchymal transformation. Here you can see the epithelial cell, epithelial rest of molasses, epithelial rests of molasses, and here some islands of this epithelial rest of molasses, and this is electron micrograph of rest molasses. These are epithelial cells, and rest the scarcity of cytoplasmic organelles and basal lamina surrounding it are notable, and also you can see the extrinsic acellular cementum, and here you can also see the cementoblast forming differentiated cementoblast, and from this side you can see the differentiated odontoblast from the pulpal side, and here you can see the epithelial cells, Hertwig epithelial root sheath, and differentiating cementoblasts. Here you can see the immunohistochemistry showing the presence of enamel-like protein at the surface of the developing root, and this represents a frozen ground section of premolar root using an antibody against amylogenin, and it is established that amylogenin is present in developing root. Some words about the cells of about the cells of connective tissue, mesenchymal origin, which such as cementoblasts, which are roughly from 8 to 15 micrometers in size, resemble osteoblasts and are basophilic. The cell nucleus is rich in euchromatin. The nuclear lamina is fibrous, and the cytoplasm contains the Golgi apparatus and extensive rough endoplasmic reticulum for protein synthesis and expert of products of synthesis. Cementoblasts are able to secrete cementum matrix in all direction, but secretion may be limited on the side facing the tooth. The cells project numerous cytoplasmic processes between which cytoplasm integrates and freshly formed collagen fibrous bundles together in this space. And this micrograph illustrates the fibroblast-like cell from a dental follicle with cytoplasmic processes lying close to the unmineralized external surface of initial root dentin layer, and here you can see the coronal termination of intact internal enamel epithelial cell of epithelial root sheath, and also here you can see the portion of odontoblasts, and here you can see that these are the cementoblasts at root surface secreting collagen fibers, and here you can see this fibrous fringe then intermingle with this unmineralized outer region of root predentin, and this is the site of future dentin cementum junction, and adjacent to it the hyaline layer. And cementogenesis, which has this, it has such stages, and here are indicated these stages: deposition of dentin along the inner aspect of Hertwig's epithelial root sheath, and breaking up of hairs, allowing the newly formed dentin to come in contact with cells of dental follicle, and this caused the differentiation of cementoblast along the external surface of the root, and protein secretion by cementoblasts, mainly collagen proteoglycans, which forms the organic matrix of cementum, and phase of matrix maturation, which subsequently mineralizes to form cementum. And there are such division of classification of cementum. It is acellular afibrillar cementum, acellular extrinsic fiber cementum, cellular extrinsic and intrinsic fiber cementum, and let's describe this each type of cementum. And you know that after embedded of cementoblast into the mineralized matrix, they become cementocytes that are derived from the cementoblast that are walled into the cementum. These cementocytes, by their own product during the course of cementogenesis. However, they are only found in ready formed layers of cellular intrinsic fiber cementum, and structurally the cementocytes still resemble cementoblasts. As they get older, the number of organelles in their plasma cytoplasm decreases. And also, you know that there are fibroblasts that are responsible for the formation of fibers in acellular fiber cementum. In terms of their structure, they resemble connective tissue fibroblasts, but may be specialized for fiber formation in cementum, and they can be found in the root region covered with acellular extrinsic fiber cementum in close proximity to the collagen fibers radiating vertically into the root surface. As I mentioned, cementum is not uniformly structured in humans. Four or even five types can be distinguished. They differ in terms of cell content, fiber, and the constituents of their collagen matrix. And there, now we discuss the acellular afibrillar cementum, which this type of cementum, acellular afibrillar cementum, contains no cementocytes and no collagen fibrils. It is the first type of cementum to be formed during tooth development. It covers parts of cervical enamel, rarely the cervical root surface. When the epithelial covering of the freshly formed coronal enamel disappears, the adjacent connective tissue comes with the enamel surface. Pre-eruptive maturation of the enamel is completed at this stage. This occurs shortly before eruption or as the tooth is just starting to erupt. At this stage, or even after eruption, cementoblasts deposit initial layers of acellular afibrillar cementum here, which is homogeneously mineralized. As this is acellular cementum, it is assumed that serum proteins and minerals precipitate together on the enamel surface without the involvement of cementoblasts. This type of cementum was formerly also known as coronal cementum. It has long been known to exist in herbivores. Those dental crowns are regularly covered with cementum even in the enamel region. In humans, this type of cementum is ultrastructurally distinguishable from the other types of cementum. However, it cannot be distinguished from acellular fibrillar cementum under light microscope or by histochemical methods. This type of cementum is also present in cemental spores and cemental pearls on the dental enamel. Cemental spores originate at the cervical margin of the enamel and overlap the cervical enamel margin to a variable extent. Apically, they merge continuously into the acellular extrinsic fiber cementum of the tooth root. In the reduced enamel epithelium is only disintegrated in patches, and the mature enamel surface therefore comes into contact with surrounding connective tissue in circumscribed areas, acellular, afibrillar cementum is laid down in the form of cementum pearls. There are found in variable number and size about 1 millimeter coronally of the cemento enamel junction. They are also found, though rarely, in the depths of fissures of premolars and molars. Acellular extrinsic fiber cementum originates pre and post eruption as a result of the deposition of cementum matrix. Most of the cementoblasts are not enclosed but shifted to the surface. Therefore, this type of cementum is predominantly acellular. The extrinsic fibers are collagen fibers that radiate from the periodontal space into the layer of cementum Scharf's fibers and anchor the tooth in the bone. This type of cementum covers from 60 to 90% of the root length as a deposition directly onto the freshly formed dentine matrix of the root. Thus, the cervical third of the teeth almost exclusively contains a cellular extrinsic fiber. Here you can see this, a cellular extrinsic fiber cementum in the apical region consists of primarily formed cellular intrinsic fiber cementum here, and here you can see a cellular cementum, and here you can see a cellular cementum and cellular cementum. The formation of a cellular extrinsic fiber cementum accompanies root development in an area within 300 micrometers of the apically advancing root edge. The zone of cementogenesis shifts more apically when the Hartwig's epithelial root sheath disintegrates and makes the freshly formed dentine matrix accessible to the surrounding periodontal cells. First, a fringe of short collagenous fibrils is deposited. Such collagenous fibrils are deposited on the surface and not yet mineralized dentine matrix. The cells that bring this about are cementoblasts, which cannot be distinguished morphologically from fibroblasts. The short collagen fibrils lie perpendicular to the root surface, their ends extended into the still non-calcified dentine matrix. Later, when they grow longer and anchor the tooth in the bone, they become periodontal ligament fibers that transverse the periodontal ligament space. In the process, the fringe of short collagen fibrils becomes denser coronally. Within 100 micrometers of the advancing root edge, fibroblast-type cells make contact with the dentine surface and start to assemble the fibril fringe into bundles that correspond to fibers. Along the second 100 micrometer more coronally, the fringe of fibers becomes even denser, and together with the cells that create it, form a dense cell fiber complex. Mineralization of the root dentine walls takes place in the fibers, then the mineralization front passes into the cementum layers where the fibers are further enclosed. Here's this process advances continuously toward the apex, about 1 millimeter coronally of the growing root edge. The acellular extrinsic cementum is only a few micrometer thick. Toward the end of eruption, before the tooth is in occlusal contact, the cementum has increased to 10 to 15 micrometer thick. Even after a tooth erupts, the acellular extrinsic fiber cementum continues to grow thicker throughout life. The Sharpie fibers in cementum are homogeneously mineralized, the majority of the crystals running with their C-axis parallel to the path of the fibrils. After disintegration of the organic components, the entry points of Sharpie's fibers are visible in specimens at the cementum surface as small round elevations with a diameter of 3 to 6 micrometer. And here you can see the cementocytes, and they lie in their lacunae, and they have such protrusions as canaliculi, and they communicate by these canaliculi. And here you can see the canaliculus and the cell which is embedded in the side in this organic matrix, and it has the nucleus, it has cytoplasm, and it lies in this lacunae. And here we can now discuss the cellular intrinsic cement. The cellular intrinsic fiber cementum, and here you can see the cellular cementum, originates pre-eruptive in the area of root furcations and post-eruptively in the apical regions. It is always the first layer of cellular mixed fiber cementum, and in subsequent layers of cellular mixed fiber cementum, it forms the intermediate layers. Cellular intrinsic fiber cementum is produced solely by cementoblasts. It contains no Sharpie fibers. The cells are distributed with variable density, cellular intrinsic fiber cementum, and in places no cells are visible. Sometimes this is acellular intrinsic fiber cementum. As in acellular extrinsic fiber cementum, the formation of cellular intrinsic fiber cementum starts with the deposition of cementum directly onto the fresh dentin layer when the Hartwig's epithelial root sheath disintegrates and thereby leaves the root surface clear. A relatively large amount of cementum matrix is rapidly formed along a zone of 100 to 200 micrometer coronally of the growing root edge, and as interweaving of matrix arises between the cementum and the dentin at the future cemento-enamel junction. And many fibers, arranged collagen fibrils, are contained in the cementum matrix, as are bundled collagen fibers, which are arranged parallel to the root surface in this type of cementum. And the cementocytes define this arrangement. Here you can see the Bible. Cementocytes define this arrangement by creating compartments running parallel to the root surface with their cytoplasmic processes. And here you can see viable superficial cementocytes. This matrix is mineralized along a mineralization front, which is roughly 200 micrometers away from the advancing root edge. The initial mineralization happens relatively quickly, and subsequent layers are formed far more slowly. In the process, layers of cellular and acellular intrinsic fiber cementum are formed. Acellular extrinsic fiber cementum may also be laid down. The cellular mixed fiber cementum originates in this way, comprising layers of acellular and cellular intrinsic fiber cementum and acellular extrinsic fiber cementum. Here you can see these acellular and cellular fiber cementum, and cellular acellular intrinsic fiber cementum are also formed in resorption lacunae and in response to traumatic tooth damage such as root fractures. Therefore, this cementum fulfills the function of repair rather than attachment of the teeth. Structurally, cellular and acellular intrinsic fiber cementum are characterized by randomly arranged collagen fibrils and bundle groups of fibrils which primarily run parallel to the root surface. There is no connection with sharpish fibers. Another notable feature is the lacunae in which the cementocytes reside, and numerous tiny canaliculi originate from the lacunae with thin cytoplasmic processes of the cementocytes running through them. The cell processes may be in contact with one another, and in the region of the cemento-dentinal junction, the canaliculi of the cementocytes may be in contact with the dentinal tubules of the odontoblast. In ground sections, the lacunae and canaliculi appear dark in transmitted light, and many lacunae appear empty in histologic sections, and this may be caused by faulty chemical fixation because cementocytes are vital cells. In addition to the described structured types of cementum, intermediate cementum has been described as a separate type, and it is supposed to be found as a separate layer between the root dentin and the cellular mixed fiber cementum. More recent studies have shown, however, that these irregular and non-homogeneously mineralized intermediate layers are not separate but represent the boundary between dentin and cementum. By contrast, the independent cementum layer, described as the hyaline homogeneous layer, lies between the dentin surface and the acellular extrinsic fiber cementum and is recognized as such. As the outermost layer, roughly 3 to 5 micrometer wide layer, overlies the acellular extrinsic fiber cementum in places, it appears this structureless under the light microscope. Here you can see, and however it is eosinophilic. Its surface lie many polygonal fibroblasts between which the periodontal ligament fibers radiate into the cementoid, and this layer, freshly made cementum, is called cementoid layer. If the cementoid surface is cleared of organic material, these fiber entry points can become visible as round elevations roughly 3 to 6 micrometer in diameter. These micrographs illustrate light micrographs, or illustrate the developing cementum and periodontal ligament of unerupted human premolar teeth. And here you can see the fiber fringe protrudes from a thin layer of mineralized acellular cementum. At this stage, the periodontal ligament consists of fibrocellular meshwork oriented parallel to the root surface, and here you can see 15 micrometer thick layer of mineralized cementum covers the root surface, and the orientation of the periodontal ligaments fibers has changed, and many are now roughly perpendicular to the root surface. Some of the periodontal ligament fibers, here you can see, are continuous with the fiber fringe. Here, this fiber fringe of this and this transmission micrograph shows the developing root fibers in the developing periodontal ligament. Here you can see these developing fibers of periodontal ligament, which are running parallel to the root root surface with little attachment to the cementum fibers, and which are perpendicular to the root surface. And here you can see the dentin cementum junction, and beneath which would lie the hyaline layer. And also here you can see the fibroblast or periodontal ligament, and the line front mineralization from extends from the dentin across the dentin cementum junction to involve the base of the cementum fibers. Here you can see this is the dentin, which is overlaid by cementum, and here you can see the sharpish fibers and the cells of periodontal ligaments, and there is no layer of cementoblasts. And here you can see these are the indicated are uh, these are the sharpish fibers, extrinsic fibers, here extrinsic fibers, and this is the decalcified root surface forming acellular extrinsic fiber cementum. And between the sharpish fibers, the cells are cementoblasts and are more cuboidal. These cells are cuboidal between these fibers, and here you can see this is the cementoblast with developed rough endoplasmic reticulum and other organelles and other complexes which are for protein synthesis. And also you can see this cellular cementum. This cell is typically more irregular in morphology and contains larger amount of the intracellular organelles. And here you can see the sharpish fibers. This is a light micrograph of human periodontal ligament. This is periodontal ligament. This is alveolar bone, and this is the cementum. And you can see the sharpish fibers extending from periodontal ligament to bone and to the cementum here. And here you can see the sharpish fibers which extend into the cementum, which are present in cementum but are less distinct. And here, these electromicrographs of periodontal ligament are inserting into cementum. Here you can see the sharpish fibers which are inserting into the cementum, and these sharpish fibers are seen in the cementum. There, here, and here they are propagating into the cementum. And here you can see this is cementoblast, and from both sides there are the extrinsic fibers, and you can see the mineralized dentin. And this section illustrates root surface showing cellular cementum, and here you can see the cementosites, and also cementoblasts. Here cementoblasts and already embedded cementosites, and also you can see the synthesized pre-cementum. And the next is, yes, this is cementum resorption and repair. This cementum sometimes undergoes resorption, and resorption of cementum by odontoblasts, similar to osteoclasts, may occur in a very variety of conditions, including during orthodontic tooth movement. And the resorbed areas, here you can see the resorbed areas may be filled in later with a newly deposited cementum, and these arrowheads indicate the dentin cementum junction. Here, dentin cementum junction. Prior to root formation, the cells of the dental follicle have the characteristics of undifferentiated fibroblasts, and containing few cytoplasmic organelles. With the onset of root formation, The cells show an increase in cytoplasmic organelles, especially those associated with protein synthesis and secretion, and extracellular spaces begin to fill with collagen and ground substances. And here you can see the synonyms for periodontal ligament, and they are periodontal membrane, alveolodentin, dental ligament, desmodont, pericementum, dental periosteum, comphosis, and so on. And the cells of the periodontal ligament, this periodontal ligament, you know that it develops from the dental follicles, and the cells of the dental follicle give rise to cementoblasts, fibroblasts, and osteoblasts of the periodontal ligament. And there is evidence that cells from the apical region of the dental papilla migrate around the epithelial root sheath and supplement the number of cells in the dental follicle. Cells may also migrate into the developing periodontal ligament from the endosteal spaces and the adjacent alveolar bone. Yes, the fibers of the periodontal ligament are embedded in newly developed cementum and the bone as tooth erupts. And here you can see the organization of the periodontal ligament fibers. And during the formation of the root and periodontal ligament, there is obvious a dramatic increase in the number of connective tissue forming cells such as cementoblasts and fibroblasts. Stem cells exist within the region to allow for this, and also and these stem cells may be they lie in perivascular location. It is not clear whether the three main types all arise from this common stem cell, or they have each its progenitor cell. And here you can see the development. At first, these developing fiber bundles of the periodontal ligament are thin and not well oriented, and here they are all directed obliquely. And after the teeth come into occlusion, the fiber bundles of periodontal ligament, well, they are now well organized in terms of thickness and orientation. And here you can see the principal collagen fibers which with an erupting permanent canine, and you can see that these fibers erupting into the oral cavity, the principal collagen fibers are seen passing from tooth to the bone, from tooth to the bone. And here you can see that these erupted teeth with collagen fibers for the fully erupted permanent canine of ferret, and these permanent canine tooth, the collagen fibers are not as well organized in thickness and orientation as those for the fully erupted tooth, which is illustrated here. The next, this is the alveolar bone develops from the dental follicle, and the mandible and the maxilla develop intramembranous. These are two ways of ossification, and this is intramembranous ossification and endochondrial ossification. And here you can see intramembranous ossification and endochondrial ossification, and whole body of mandible except the anterior part is undergoes the intramembranous ossification, and ramus of mandible as far as mandibular foramen also. And the anterior portion of the mandible symphysis, part of the ramus above the mandibular foramen, and coronoid process, condylar process, they undergo endochondrial ossification, and we discuss it now. And this alveolar bone, as we mentioned, they are developed from dental follicle, and you know that dental follicle These ectomesenchymal cells of the dental follicle differentiate into osteoblasts and lay down the matrix called osteoid. This is unmineralized part of the bone, and some osteoblasts become embedded in the matrix as cementoblasts. You know, these osteoblasts also are become embedded in the organic matrix, and after mineralization, they are called now osteocytes. And the mandible and the maxilla develop intramembranous ossification in the fibrocellular condensation and center. Here you can see how this takes place. This is intramembranous ossification. You know that this condensation of mesenchyme, this is mesenchyme, and the condensation, a center of ossification appears, center of mesenchymal, the condensation mesenchyme and center of ossification appears, in which osteoblasts lay down first form embryonic or woven bone. As the teeth develop, bone extends from the developing mandible and maxilla to surround and protect the teeth, and forming of alveolus. This alveolus is separated from the developing enamel organ by the dental follicle, and to accommodate the growing tooth germs, the lamellae of this developing alveolar bone undergo resorption on the inner wall of the alveolus, and while on the outer wall of the alveolus bone is deposited in, and the developing teeth therefore come to lie in a contact of bone, and later the teeth become separated from each other by the development of interdental septa. With the onset of root formation, interradicular bone develops into multi-rooted teeth. As in other sites, the collagen fibers in the newly formed alveolar bone have a more variable diameter and lack a preferential orientation, and this immature woven bone is subsequently converted to fine fibered adult lamellar bone. The source of the cells forming alveolar bone is uncertain, although some have suggested that it may be from neural crest cells of the investing layer of the dental follicle. And here, this is high magnification. You can see this bone, and this is the condensation of mesenchyme, and you see that these osteoblasts, the secreted matrices of the bone, and the surfaces of the which remain covered by osteoblasts. This you can see, this osteoblast between these thin regions of new woven bone are areas with small blood vessels. Yes, here you can see blood vessels, and it is high magnification, and you can see the ossification centers of this mesenchyme. And here it is intramembranous ossification, when ossification centers fuse to form these trabeculae, and blood vessels grow in between these trabeculae and these gaps between these trabeculae. And endochondral ossification, the other way of ossification is endochondral ossification, in which the pre-existing matrix of hyaline cartilage, you can say this is hyaline cartilage, is eroded and invaded by osteoblasts, which then begin osteoid production. And here you can see these are the remnants at the bifurcation of roots. You can see these are enamel pearls, and these enamel pearls appear as spherical masses, and they are developed from Hartwig's epithelial root sheets. These are the remnants of Hartwig's epithelial root sheets, and develop ectopically in the area of root bifurcation or other sites. And these are These are the small isolated spheres of enamel that are occasionally found at the root surface, and they are particularly common in the root bifurcation area, and but may also occur at lower levels, usually in interradicular region. And here you can see odontohypophosphatasia, and this is the condition in which there is reduced activity of tissue, non-specific alkaline phosphatase, and it is a rare autosomal recessive disease in which there is a marked deficiency in the presence of both acellular and cellular cementum. The lack of attachment of periodontal ligament fibers to the residual cement is associated with premature loss of the deciduous tooth and teeth. In such patients, any orthodontic treatment is likely to be accompanied by the premature loss of any permanent teeth required to be removed. And also here you can see the periodontal disease, which is most important clinical condition affecting the periodontal ligament, is chronic inflammatory periodontal disease, in which toxic products are released by dental plaque lying in the gingival crevice. And interesting with the host the defense mechanism, including gingival crevicular fluid. These products result in the destruction and the loss of periodontal ligament tissue and adjacent alveolar bone. Such a process leads to the formation of periodontal pocket, allowing for the accumulation of more plaques, and vicious circle is established. The loss of attachment tissue may expose the root in the mouth, and with increasing mobility, the tooth may eventually be lost. And here is also the Epstein-Barr virus, which affects over 90% of humans and is usually transmitted by oral secretions or blood. And the virus that replicates in epithelial cells or B cells of the oropharynx. And the most common symptoms of infectious mononucleosis are fever, lymphadenopathy, and pharyngitis, and oral ulcers, palatal petechia, and less commonly gingival ulcerations. Thank you.